So the functions of phospholipids are best understood if they are learned component wise. So in the previous videos, we learned that phospholipid is made up of lipid bilayer, is a lipid bilayer, which is phospholipid bilayer, which is these things that I'm drawing. These these molecules are called phospholipids because they are present in two layers, the one above, the second below. The whole structure is called a phospholipid bilayer. We also learned that a phospholipid is made up of a phospholipid is made up of polar heads. These these blue heads are polar, meaning that they can attract polar compound, whereas these yellow tails mm, these yellow tails are non-polar, so they attract each other. So what happens is this region which is formed in the lipid bilayer, this region, because hydrophobic tails are present in this area, this hydrophobic meaning that it's water hating. So basically, no polar molecule can pass freely from outside to inside of the cell or from inside to outside of the cell. From inside to outside of the cell. Why is that? That's because although it would be the polar molecule would be attracted to the polar heads, that is that it would be attracted to the polar heads, but it won't cross the cell membrane. Why? Simply because this hydrophobic region will be repelling it. So just because of this property, that the phospholipid bilayer is partially permeable membrane because it allows non-polar molecules like oxygen or carbon dioxide to easily diffuse through. These things have absolutely no problem crossing the cell membrane freely, whereas polar compounds like glucose or amino acids don't they generally tend to not cross the cell membrane easily. So which begs the question, well, if glucose and amino acids, these are two pretty important things which are needed by the cell for its survival. These are crucial for its survival. So how do these things pass the cell membrane? Well, it's because remember the last video I taught you that there are basically proteins in the cell membrane. They are divided into two groups. One of them being these are external proteins, which I'm highlighting right now. Sorry, um, extrinsic proteins. These are extrinsic proteins, whereas the red ones are intrinsic proteins. Now, these small these two are half span half spanning intrinsic proteins, whereas is this one is full span intrinsic protein. Now, what happens is that these full span intrinsic protein, they generally, or most of them, are transport proteins. Although they have other subtypes and they have perform other functions too, but most of them are transport proteins. I'll talk about this in detail in later lectures, but essentially what you need to know right now is that these the so these proteins have a channel, essentially a channel running in between them, and and the amino acids surrounding these channels are polar. Whereas obviously this area, because it is embedded in the phospholipid bilayer adjacent to the hydrophobic tail, these amino acids are non-polar, so that it remains stuck to the phospholipid bilayer. So what happens is that because this channel is surrounded by polar amino acids, Polar compounds like glucose and amino acids can diffuse through in or out can diffuse through these proteins. This is called facilitated diffusion because the diffusion of glucose or amino acid is facilitated by this protein. So what I've been basically trying to tell you is that for a cell to survive, it is crucial that it controls that it controls the contents of its cytoplasm, right? Because there are different biochemical pathways, different reactions going on over there. So it's essential for it to control a certain concentration of different compounds in its cytoplasm. How does it do that? It does that by restricting entries of certain molecules because of phospholipid barrier, which is partially permeable, but when it needs polar compounds, it can get it through these proteins. Okay, so there are other functions of these proteins and these lipids as well. You can see that I have drawn these things. These things are attached to phospholipids and proteins. These things are small carbohydrate chains. And what this essentially means is that if they are attached to a protein, then this will be called a glycoprotein. If they are attached to a lipid, these will be called a glyco glycolipid. Now, the main function of these two glycoproteins and glycolipid is that they act as receptor. So a cell, in order to survive, in order to no, in order to respond to a certain stimulus, it needs to detect a molecule, a signaling molecule. Receptors detect that signaling molecule, and these also they also you might have heard that your blood group is A or B, B positive, B negative, A, B, O. Yeah. S- So you might have heard about your blood groups A, B, and O. What these basically are different glycoproteins. So A is a different glycoprotein, B is a different glycoprotein. This is a way of your red blood cells to basically identify themselves as your own, so that your body can detect whether there are foreign cells present in the body or not. And you can also imagine that these glycogen molecules or glycoproteins and glycolipids form a sort of, if this is a cell membrane, they form a sort of covering over the cell. This covering is called a glycocalyx. And what it does is that because carbohydrates are polar, this attracts water and this gives stability to the cell. Okay. Now, one last thing. These little white molecules that I have drawn over here, these little white molecules, these are you might remember them them as cholesterol. I told you that cholesterol has a polar head and a non-polar tail. So the main function of cholesterol is to basically regulate the flow of cell membrane. When it becomes too hot, basically what hot becoming too hot means that it's the phospholipid molecule, these lipids and proteins start to move more rigorously. They have more energy, they have more kinetic energy, so they start to move around more rigorously. What it does is the polar head, because it's polar, it attracts the other polar compounds, as in other polar heads of phospholipid, and it tends to bind them, and it tends to bind the phospholipid together. Whereas if the temperature is too cold, this tail will prevent the phospholipid membrane becoming too less fluid. So in the next few lectures, I will explain how facilitated diffusion and how